Hello. My name is Ross Kedl. I am a professor of immunology at the University of Colorado Anschutz Medical Campus. And today I want to talk to you about adjuvant elicited T-cell responses for therapeutic intervention. My lab has been very interested in cellular immunity or CD8 T-cell positive immunity for many years now, and very interested in seeing how big of an immune response we can generate for the purposes of therapeutic intervention in chronic infectious diseases and cancer. So I assembled a few goals for the purposes of the talk. I'm not going to read through them, but suffice it to say that hopefully at the end of this, I will have achieved those goals. You can be the judge of that. My graduate advisor was Matt Mesher at the University of Minnesota. And one of the things he told me when giving a talk is that when you give a talk, you tell people what you're going to tell them, then you tell them, and then you tell them what you told them. So I'm going to follow his advice because he was a very wise man. And first of all, give you the answers to everything. First of all, the questions that I'm going to try and address, and then secondarily, what I believe to be the answers. And then we'll step through each one, and I'll try and convince you of those particular answers. So a major question, first of all, is what is the capacity of the immune response to respond to vaccination? How many T cells should we be able to make by that? And I think the data are pretty consistent that a significant immune response, double-digit percentages, that out of the total CD8 or T cell pool, double-digit 10% and upwards of the total T cells we should be able to generate following vaccination. That's a pretty good number because the number we need for therapeutic efficacy is certainly in the millions, if not billions range. So we need a lot. Many T cells are needed for therapeutic purposes. So we think that the immune system should be able to handle that. Our current adjuvants, frankly, don't measure up all that well. They're at least 10 to 100-fold off when you look at the frequency of cells in the peripheral blood. There may be some benefit if you look in the tissue that may offset that. Despite that low frequency, there are some things that are working or seem to be working a little bit. It's a little hard to say for sure, but they certainly are consistent with the idea that these vaccines are working. But we can certainly do way better, and there are a handful of things that our research over the last 20 years has revealed as significant influencers of the way in which adjuvants elicit cellular responses. And they have some distinct characteristic differences from how adjuvants elicit humoral or antibody-based responses on which most adjuvants are selected. Most are picked and developed for the purposes of prophylactic or preventative immunity against infectious diseases. None of them are developed for the purposes of cellular responses. And we're going to have to take a few different approaches if we're going to make that work better. And there's a handful of things that we'll discuss that I think are important to consider when trying to increase the efficacy of them. Okay, so how many T cells should we be able to make? What's the capacity of the immune system to make T cells downstream of the vaccine? And it may be worth just for a second to step back a little further, just say, well, do we really even need T cells? Is that important? Antibodies are a very important part of the immune response. Aren't they good enough to get rid of cancer? There's a lot of other immune cells going on. What's the evidence that T cells are really necessary for therapeutic efficacy? And the new immunotherapeutic modalities that have been out for the last 10 years or so, what are known as broadly as checkpoint blockade, antibodies that cut off an immune checkpoint and let the immune system run more vigorously. The overwhelming data from all of those studies is that those immune modulators work, and when they work because they make lots of T cells. And if they don't make T cells, then they don't work. So here's some examples of that. You're free to look up those references, and I highly recommend it. But broadly speaking, I think everybody can reasonably conclude that therapeutic efficacy against cancer can actually be achieved. And when it is achieved, a therapeutic efficacy through immune modulation is possible to achieve. And when it is a T cell dependent phenomenon. Okay, so given the fact that we need the T cells in order to elicit this therapeutic immunity, how many can we make? The yellow fever vaccine serves as a really good gold standard for most people's estimation. Now, this is a live attenuated vaccine. It is not actually an adjuvanted subunit vaccine, which is really more where the world of vaccines is moving, and my talk is centered primarily on adjuvant elicited immunity. But this at least lets us know what's the capacity of the immune system. And Rafi Ahmed's lab has done a lot of the pioneering in this regard. And here's a couple of papers of his that show sort of what tracking the immune response after the yellow fever vaccine looks like. And generally speaking, you can get into the upwards of 10%. Not everybody does. Most people get into the 5% range, but people can certainly get to 10 to 20% of their T cells can be directed at the vaccine itself. So if you do sort of a back of the envelope calculation as to how many PBMCs we have in our blood and how many liters of blood we have, and the frequency of those that happen to be CD8 T cells, which is primarily what I'll be centering this talk on, is for cellular immune purposes. You come up with a number of T cells that if you had 5 to 10% of your antigen-specific T cells in PBMCs, this would be somewhere between 10 and 30 million. So that's a very large number. There's evidence that we can, other infections can get us even higher. The higher you get on that number, the more you can have some immune pathology. Making that many T cells takes a lot of energy resources, and that can have consequences. But the immune system certainly has the capacity to make in the tens of millions of T cells for the purposes of a therapeutic response. So we know we have a pretty good ceiling to shoot for. How close to that ceiling do we need to get? Maybe we can get there by a fraction of that. Maybe therapeutic efficacy comes as a result of much fewer T cells. A number of years ago, Sam Silverstein had a very important paper, at least in my view, in which he asked that question: how many T cells do you need? Broadly, at the time, it was believed that T cells are serial killers. When a T cell kills a target, it kills one, then it can detach and move on and kill another. And so it's one of the reasons they were thought, oh, T cells are great, right? They can, you don't need very many of them, and a single T cell can take care of 10 targets or so. And it turns out that they're more, T cells are more hand-to-hand combat, mano-a-mano, than they are 1 per 10 targets. In these experiments that he did, he could transfer T cells and tumors for which the T cells were specific, and ask the question, how many T cells did he need in order to control that tumor growth? And you can see it being controlled at 10 to the 6th T cells per mil. So 1 million T cells per mil of blood was what it took to control the tumor, and 10 million per mil it took to actually make the tumor go away. And his conclusion was broadly that 10 to the 7th T cells per mil, or per centimeters cubed if you're looking at tissues, is required to produce sterilizing immunity. And there's a number of publications since then in which they've evaluated patients whose tumor is melting away or going away as a result of immunotherapy. And when they look in that tissue and count up the number of cells within the tumor tissue, it's within very much striking distance of this limit that Sam established so many years ago. As a side note, this frequency of T cells in the blood is actually considerably less than that, which Sam identified as a couple of logs less, or almost, that Sam identified as critical for tumor regression. Now, some of that is going to probably be offset by the fact that the T cells can accumulate within tissues, and the tissue concentration can go up even though the blood levels may be a little lower. So some of that could be offset by that caveat. But essentially, we definitely need into the tens of millions, if not billions, of cells in order to get therapeutic efficacy. So this is a high bar to reach, obviously, especially given what so far we've demonstrated with our adjuvants, or with the vaccine so far. So with that in mind, how close are our current adjuvants to achieving that particular goal? This is a compendium of a number of studies that have been done, a lot of them recently through the COVID vaccines, but some going back many years, with various adjuvants and adjuvant platforms and vaccine platforms. And what you can see in the blue numbers on the right is, broadly speaking, they all come to the same conclusion, which is you tend to get about 0.1% of the total T cell pool is you can capably generate that from pretty much any of these adjuvant platforms. Sometimes you can lean into the 1% range, but in the blood, it's really difficult. None of these adjuvants have demonstrated the ability to make the frequency of T cells like the yellow fever vaccine, and certainly not like seems to be necessary for therapeutic efficacy. If you look down at the bottom of the slide on the right, you can see that if you look in the lung tissue, as I mentioned before, Some of this may be offset by the fact that the tissues can concentrate the frequency of T cells in terms of the concentration of them per per centimeter cubed of tissue. And the lung is a good example of that. And so we've been involved in some of these studies down below and can see a really high frequency of cells accumulating in the lung, and that's going to be beneficial, significantly beneficial in achieving this number. But broadly speaking, our adjuvants are at least 10 to 100-fold off, especially when you look in the peripheral blood. And hopefully the tissue evaluation offsets some of that. But our adjuvants are not measuring up spectacularly. Given that, really, is anything working at all? Is there any successes that we have? And actually, surprisingly, to me at any rate, there are a number of examples in the recent literature in which groups are taking are sequencing tumors and asking the question, how many mutations does the tumor have in self-antigens? And then they're creating vaccines centered around those mutated epitopes. The idea being that they're mutated from the regular sequence, and so they should potentially serve as a neoantigen against the tumor. The immune system won't be selected against it. It's going to behave more like a viral antigen of some sort. So they collect all of those, they package them into various vaccines. mRNA vaccines are very popular for this. And then they begin a process of intense vaccination of the patients. And you can see here it says up to 12 vaccinations and continued treatment. So it's a non-trivial vaccine regimen, but you can also see that immune responses to these neoepitopes, these mutated epitopes, can be generated to a pretty decent number. These are not, some of these are pretty high numbers, but these are not anywhere above 1% of the total immune response, but collectively they add up and they seem to be doing some useful things. In this particular study, they were doing essentially the same thing. They were identifying these mutated antigens, and then instead of using mRNA, they were using, they made synthetic peptides, and then used poly-IC as an adjuvant, and again, a intensive sequence of priming and boosting. And in a number of patients, you could see some decent T cells being generated. Some of them in the single-digit percentages, but again, the vast majority under a single percent, but detectable, they're there, and even providing some clinical response that you can see down here in a couple of patients that had detectable immune responses. Generally, if there's detectable T cell responses, the patients tend to do better. And here's a good example of that most recently, looking at responders and non-responders and how progression-free survival is being achieved over a couple of years from those who are responding, compared to those who are not. And again, these are really intensive vaccine regimens. But worth noting, there are as many non-responders as there are responders. So even though the responders, they're not making very large T cell responses, they do seem to have some efficacy. At least as many people aren't responding at all. So it's fair to say that even though there may be some hints at successes, we can clearly do an awful lot better. So the question is, what are we missing? And this is where my lab has spent most of the last 20 years trying to really understand the fundamentals of adjuvant elicited immunity. What are the rules guiding how adjuvants generate immune responses, and can we manipulate those rules to our benefit? So before we get into that, just to make sure we're all on the same page, T cells don't get activated by soluble antigen. Antigens don't show up and stimulate a T cell directly. T cells actually have something called a T cell receptor. And that T cell receptor is specific for a particular peptide antigen that is loaded into molecules called major histocompatibility, class 1 or class 2. CD4 cells interact with peptides in the context of this class 2 molecule, and CD8 T cells interact with this class 1 MHC plus the peptide. So the way they interact with that is through the action of an antigen presenting cell, which, as its name implies, does the heavy lifting of presenting antigens to the T cells, essentially allowing the T cells to scan the surface of that APC. And if it's specific for a particular antigen on it, it initiates the process of immunity. So a common way that this is conceived of is that a T cell, a CD4 T cell, is capable of interacting with this antigen presenting cell if its TCR is specific for this particular peptide bound to MHC class 2. T cells are notoriously insecure. They need constant reaffirmation and support. And so they're always looking for co-stimulatory molecules. A chief one in both CD4 and CD8s is this molecule called CD28 that receives, that's ligated by its receptor or ligand that's on the antigen-presenting cell CD8086. Upon receiving that signal, along with some cytokines that I'll talk about in a second, the CD4 T cell actually upregulates some activation molecules of its own, which then in return stimulate the dendritic cell to undergo a process of expressing other molecules that are really important in generating CD8 T cell responses. And here's some examples of that. And this is a process known as licensing of the antigen presenting cell. Up until this licensing process, that T cell is less capable of generating the CD8s, but after interacting with the 4s, it makes much better CD8 responses. And this is, it used to be thought that CD4 T cells provided help to CD8s by IL-2, and that's largely been dispelled. Almost all of its co-stimulatory or helper activity in generating CD8 responses is really through cell surface receptors and activations of APCs, along with some other inflammatory cytokines. So upon productive interaction of all these molecules, there's also a lot of cytokines that are produced, soluble mediators of inflammation. T cells make some, APCs make plenty, and all of those can provide co-stimulatories to the CD8 T cells and CD4 T cells. They undergo a rapid round of clonal expansion. They divide once every 2 hours or so if it's a CD8 T cell for many days, and 10 cells pretty quickly become 10 million. And then they go off, or if you're a CD8 T cell, you go off into the world and find a target to kill. If you're a CD4 T cell, you go off into the world and help other things orchestrate immune responses of a variety of kinds. Importantly, all of these inflammatory signals can be, in one way or another, initiated or elicited from an antigen-presenting cell through the use of innate stimuli or inflammatory mediators, or also what we call adjuvants, which are the inflammation instigators we stick into a vaccine. Because if you get rid of any of these molecules in the process, the entire T cell response tends to collapse pretty quickly. So these inflammatory mediators are a necessary precursor to ultimately creating the adaptive response. And so this boundary between the innate system and the adaptive system is where we've spent a lot of time thinking about. And a lot of us really thought that this was going to be pretty easy once we identified innate stimuli and innate receptors that could generate some of these molecules. We thought, well, this is going to be great. We're just going to throw these inflammatory stimuli into our adjuvants, and we'll generate these phenomenal T cell responses. And generally speaking, that's not unreasonably true for antibody responses. It's not that hard to get antibodies produced comparatively to cellular responses. And when you add inflammatory agents to a vaccine formulation, you generally get better antibody responses. So this is an example from Raul Torres in our department. And I've superimposed a couple of inflammatory stimuli or adjuvants that we can use, CpG or TLR7 agonists. And if you look at the antibody response without these adjuvants, it gets better no matter which ones you add. Unfortunately, the same is not true for cellular responses. We can take, and this is a method called MHC tetramer staining, and for the purposes of this, just look at the number of dots inside the circle. And you can see that when we add these inflammatory components or adjuvants, we really don't see that big of an improvement. We don't get that many more T cells, especially when you compare what you can get from an actual infection in the mouse. So our adjuvants are pretty good at augmenting antibody responses, but they're not very good at augmenting cellular responses. And a broad conclusion you can make from all these studies is that making 10 to 50,000 lymphocytes, be they T cells or B cells, it's not that hard to do. Most of these adjuvants, if you look at the frequency of B cells that you see in A, it tends to be somewhere between 10 and 50. And if you added them all up in what you see down below for the T cells, it's about that as well. Thankfully, 50,000 B cells really work well for providing antibody-mediated protection. Each B cell can crank out milligrams of antibody on its own as a plasma cell, and the system can be protected against things for which antibodies are successful. But unfortunately, 50,000 CD8 T cells rarely did anyone any good. We just, as I discussed previously, we need to make a lot more of them. So the question is, if simple adjuvants and simple approaches that make antibodies successful don't work for cellular responses, what may work, what may be better, what factors do we need to take into account? So there are a number of sort of physical, chemical, structural approaches to vaccination that we've sort of overlooked, mostly because again, almost all adjuvants are developed for the purposes of successful B cell responses. And the rules are very different between B cells and T cells in terms of what sustains a T cell versus what sustains an antibody response. So the first example of that is creating an antigen depot or an emulsion at the injection site versus having antigen delivered more solubly. For antibody responses, one of the first rules of adjuvants was long ago with Glennie back in the 1920s, was to make an emulsion of some sort or a precipitate that would stick around within the tissue for a very long period of time, what's known as an antigen depot, and would slowly drain antigen from that site into the animal and maintain the antibody response over time. And Shane Crotty and others now have shown that it's very true that if you have an antigen depot that drains antigen into the local lymphoid tissue, over the course of a month or two, you get better and better antibody responses. Unfortunately, it really hamstrings your T cell responses. And here's an example of that. We can formulate the adjuvant that I'll discuss in a minute in solubly in PBS. And when we inject it, we have a very nice high frequency of T cells in this mouse model. All we need to do to destroy its efficacy is to put it in incomplete Freund's adjuvant. So we make an emulsion out of it so that the adjuvant itself and the antigen is stuck within the tissue for a long period of time. And it's almost as bad as if we never added adjuvant. Willem Overwyk, a number of years ago, actually nailed down exactly why this occurs. And I won't go through all the details of this, except to say that he could create both a tumor site and a vaccination site. And if he used incomplete Freund's adjuvant, all the T cells generated, instead of going to the tumor, they all went to the vaccine site where all the antigen was still sitting in there in the emulsion. And they died vigorously trying to get rid of that antigen. But if he injected the antigen solubly, now all the T cells instead went to the tumor. So the formulation of the adjuvant can take the most spectacular adjuvant in the world and turn it into the worst. And so antigen depots are a significant problem for generating cellular immunity for therapeutic purposes. Particularly since so many of our adjuvants that we generate continue to be formulated in emulsions or in precipitates. So related to that, a lot of those adjuvants are formulated in a way and stimulate receptors that create tremendous amounts of inflammation. So this is some research from my lab where we're looking at our combined adjuvant that I'll talk about more in a second. And we can add another molecule called CpG to this. And a number, this is just a small set, but a number of inflammatory factors go way up when we add this additional inflammatory signal. What's really curious about that is when we add that inflammatory signal, we actually make more T cells, but what we do is we make a lot of short-lived T cells, not a lot of long-lived T cells. And in fact, when we add more and more of this inflammatory component, as time goes on, what we find is we compromise our long-term immunity. We begin stealing from our memory pool and destroying them as short-lived effectors instead of maintaining a long-term, long-lived T cell memory. And of course, the point of adjuvants and vaccines, broadly speaking, is immune memory that can be utilized later. So if our adjuvants are too inflammatory, we can make a spectacular primary response, but we really compromise the durability of that response over time. And again, so many of our adjuvants, they're all developed for the purposes of antibody responses, and all of that inflammation actually makes the antibody response go up and up and up and up, and it makes the T cell response go down and down and down. Lastly, Bob Cedar has done a lot of work in this space, and this is a really excellent example of his work using a vaccine that is made, that's formulated as a nanoparticle with agonists for the toll-like receptor 7 and 8 pathway. And the major take home from this is that he can deliver this vaccine two different ways. He can deliver it subcutaneously or intravenously. And when he does subcutaneously, you see the magnitude of the immune response in the blood is higher than that when he delivers it intravenously. But what ends up happening is that those cells, much like the previous slide I showed, where when you add more inflammation, you end up with a lot of short-lived effectors. In the sub-Q setting, you get a lot of these short-lived effectors in the blood, whereas the intravenous delivery actually gives you more of the memory cells. And because it has more memory cells, when you look at their long-lived efficacy against an implanted tumor, The subcutaneous vaccination has very little efficacy, but the intravenous has significantly better. So the route of delivery, we've almost always been obsessed with subcutaneous and intramuscular, but for cellular purposes, intravenous delivery of vaccines is almost certainly going to need to be approached and attempted to really get the numbers of T cells that we need. Okay, so those are some sort of practical ways. What about biologically speaking? Are there biological pathways we can address or be more intentional about besides just injecting IV versus subcutaneous and not making antigen depots? In our work, we've spent a lot of time looking at combined adjuvants, and here's a good example of that where, and again, if you just looked in the circle for the number of dots, that's the number of T cells we successfully made. We can use a couple of different adjuvants, in this case, the CD40, an agonist for the CD40 pathway, as well as this inflammatory stimuli called poly-IC that generates a lot of inflammation. Either one of those will give us a certain number of T cells, but when we put the two together, we get this synergy between the two, right? So 1 + 2 = 30 instead of, it's not additive, is the point. So combining adjuvants reasonably and thoughtfully and systematically can give us a whole that's much greater than the sum of the parts. And one of the ways that this happens, we know, is that it actually increases the antigen presenting cells' ability to present antigens to CD8 T cells, something called cross-presentation. So that's a major strategy, is targeting. This specifically targets the antigen presenting cells. Neither of these agents target the T cell itself. They all target the antigen presenting. Most inflammatory agents do, but these are sort of synergistic targeters of that. So combined adjuvants we think is really important. Another way you can target the antigen presenting cell is conjugating the stimulus against the antigen presenting cell with your antigen of choice. So if I take and I link my antigen chemically to either of these, I get a much better response on both cases. So both TLR agonist antigen linked as well as antibody linked antigens. And then, as I showed in the previous slide from Bob Cedar, nanoparticle technology is a very important thing that packages everything into one package and delivers it to antigen presenting cells. The mRNA lipid nanoparticles work significantly along these ways when they elicit cellular immunity. Okay, so targeting the antigens to the APCs and enhancing their function is important. And then lastly, the two pathways that we think are very important to be much more thoughtful about as we develop better adjuvants are what we just call the 27s. We think it's all about the 27s. And by that, I mean a surface molecule called CD27, and then a soluble molecule called interleukin 27. So CD27 is, I showed you before that it's a T cell, it's a molecule that's on T cells, and it interacts with a molecule called CD70. So one of the questions we asked many years ago was, we know that this combined stimulus is so clearly so much better than each individually. So the prediction was, well, the double stimulus must do something unique to the dendritic cell that single stimuli doesn't do. And when we looked at what it did, we found that a couple of molecules were uniquely upregulated really robustly. These are two different subsets of antigen-presenting cells. A couple molecules were uniquely upregulated on these antigen-presenting cells, and only when we used the combined stimulus, not when we used either one alone. And so this suggested that this molecule, CD70, was stimulating on the T cell, CD27, and that was having a really substantial impact in this synergistic influence and ability of this adjuvant to synergistically expand the T cell pool. And sure enough, if we actually take mice that have been immunized this way, that have antigen-presenting cells that express this molecule, and we use an antibody to block that, we can actually take that immune response and neutralize the vast majority of it by simply blocking one single molecule on the APC from interacting with its receptor on the T cell, CD27. And so most adjuvants are not screened for their ability to elicit CD70 on APCs, or the expression of any of these molecules. They, broadly speaking, are not utilized as screens for better adjuvants. And in our view, they should be focused on significantly. Oops. And I should say that we've been working in this space for a very long time, for the last 20 years, but there's a whole host of people, including Ralph Steinman, who's passed away, but was a Nobel Prize winner, as well as a host of other very notable internationally recognized individuals, all would concur that CD70 interacting with CD27 may be one of the linchpins for launching T cells into the stratosphere. Lastly, interleukin-27 is another one that we discovered about 10 years ago. It has a really unique influence on adjuvant-elicited immunity, but not on infectious immunity. So when you take and immunize mice with the adjuvants we have, we get very robust immune responses that are, if anything, considerably better than an infectious agent in this model system. But if we get rid of the ability for the T cells to see IL-27 or receive signals through IL-27, I just realized that says IL-127. That should say interleukin-27. That really compromises the adjuvant-elicited response, but it has no influence at all on the infectious elicited response. So you can see that quantified over here. Response to vaccinia could care less whether or not there is or isn't IL-27 in the system, but the adjuvant-elicited immunity takes a major nosedive. And that's true no matter what adjuvant you use. It doesn't need to be dual adjuvants, it could be single adjuvants. All adjuvants, as far as we can tell, operate on this principle of needing to see IL-27. And that actually includes the mRNA-based vaccines. We now have evidence that mRNA vaccination of the type that's in the COVID vaccines, also in order to elicit CD8 T cell responses, is necessary to generate T cells from that particular vaccine as well. So I just realized I left the sentence in here. We've actually got some mechanistic understanding as to why IL-27s, both of them are needed for it. And basically, they're both needed. T cells can get off the ground, but they need to have sustained proliferation over a long period of time. And if you get rid of either of these things, T cells can start proliferating, but they can't keep doing it for 7 or 8 days, well, 6 or 7 days. So both of them are needed for the sustained, durable commitment to cell division, specifically to adjuvants. Infections use other signals to maintain durable proliferation, but in adjuvant settings, no matter the adjuvant, they need to see IL-27, otherwise they're not going to get off the ground. Okay, so that is the broad conclusions, both questions and answers. Hopefully they have been convincing for you. And if you are in any way, shape, or form involved in adjuvant screening in the future, I would like to draw particularly your attention to this last part. Because so much of the world is focused on adjuvants that elicit antibody responses, very few people sort of think much about these things, but we really need to devote some larger efforts at making adjuvants capable of generating robust cellular responses for therapeutic purposes. I'd like to take a second to thank my laboratory. A lot of people were involved in this. All of them are shown here. This happened to be a day when randomly so many people wore a dark shirt and green pants, and we just felt like we had to capture that. I have a number of very important collaborators. Chris Hunter has been a long-term collaborator in the IL-27 story, and we have some very important funding that lets us continue doing that. Also, Randy Noel at Dartmouth and Bob Cedar are people I've been collaborating with for many years, and much thanks to them for the collaboration. So thank you very much. Hope this was informative, and thanks.